name is Nghi, and I'm presenting on the hemodynamic and genetic contribution to aneurysm pathology and treatment outcome. I'd like to thank the Neurological Surgery Summer Student Program, as well as Dr. Alicia, Dr. Levitt, Dr. Kim and their labs. I'd like to give a special thank you to Sari and Corey Kelly for their guidance through this program. Background on cerebral aneurysm, it is a weakening of the artery wall in the brain. There are several factors that contribute to aneurysm pathology, including hemodynamic changes and genetics. There's also research that's shown that unruptured aneurysms in families with 2 or more members with aneurysm is as high as 18.1% compared to the 3% in the general population. So with that in mind, we have hypothesized that different areas of hemodynamic stresses in cerebral aneurysm directly drive the quantifiable expression in key vascular remodeling factors in aneurysm endothelial cells. The final product of our research is to demonstrate the link between hemodynamic stresses and endothelial cells' response when exposed to those stresses. This will enable us to bridge the gap between computational fluid dynamics and vascular biology. To do so, we have established 2 aims. 1, creates patient-specific CFD simulation and 3D printed models of cerebral aneurysm. 2, quantify the relationship between hemodynamic forces and endothelial expression of key factors implicated in aneurysm pathology. This is made possible by starting with the intraoperative imaging. These images are then processed through virtual modeling. They are then used to 3D print aneurysm anatomy and then seeded with endothelial cells. Simultaneously, we also run a computational fluid dynamic simulation, which generates a heat map of hemodynamic areas of interest, where we then extract the cells and perform genetic analysis. We begin the study in the clinical side, so we begin in the angiography suite. We utilize 3D rotational angiography to obtain patient-specific anatomy. We then use the Philip combo wire, as you can see below, to obtain patient-specific measurements of blood pressure and blood velocity, as you can see on the right. And this is done in 4 locations near the aneurysm. These measurements provide a boundary condition for the computer simulation. Here is CFD, one of the computer simulations. It stands for computational fluid dynamics. It is traditionally a mechanical engineering field of study, studying the movement of fluid. Here, we're studying cerebral blood flow. We use patient-specific boundary condition to conduct blood flow simulation using supercomputers to solve the Navier-Stokes equation. Our outcome is wall shear stress gradient, and flow. Wall shear stress is the stress the endothelial cells experience when blood flows by. When there's a change in blood velocity, this creates small oscillation or vortices that then increases the likelihood of vessel wall pathology. And another outcome of CFD is it generates a heat map of hemodynamic regions of our interest. And then we're going to move on into virtual models. I'm going to be focusing on these a bit more because this is what I've been focusing on. We have 2 programs. One is MeshLab and the other is SolidWorks. MeshLab allows for editing triangular mesh, converting from a hollow body into a solid body. We then use that STL file and put it into SolidWorks, where we then extrude some parts and cut other parts before printing them. Here I have 2 printed models, which I'm going to pass around. This one is the SLA, and this one is the FDM. So the 3D printing part, the top is the resin, and the bottom is, top is the resin, bottom is the filament. We then put it in an intermediate mold, put a soluble wax in it, and then cast it in silicone. Here we start with our SLA print. We are then going to sand it to make it for a smoother model. We're then going to coat it in XTC 3D. XTC 3D is a liquid which makes the model smoother and much more shinier. We're then going to degas sort of clear 12A and B, and this is for just clarity reasons. We're then going to pour the liquid into your intermediate mold, and this is what we have. Here we have started cutting the intermediate cast. And then here we have Mohammed, one of the students, just pouring the soluble wax in. Here I'm taking the soluble wax out, and for every patient-specific aneurysm, we create about 20 to 30 because we tend to break them in the process. And we're experimenting with different colors so we can see them better. Now, this is making the final box for the next process. Here, I'm going to degas the silicone so we can later pour it into the box. And here's our final cast. So with the final model, we're going to seed it with general carotid human endothelial cells, put them under patient-specific flow for 24 hours, use CFD hemodynamic map to determine the location for the biopsy. We're then going to conduct genetic analysis with single-cell qPCR. The results that we found was that the models were successfully populated with endothelial cells, and they survived under flow for 24 hours. We also found that there was disorganization, morphology in the aneurysm dome, decrease at atom TS1 and NLS3 expression, correlates with area of substantially lower wall shear stress and wall shear stress gradient in CFD situation. So in conclusion, creating 3D printed models of patient-specific aneurysm and populating with them with endothelial cells is feasible. An analysis of these cells flowed differences in cell morphology and genetic expression, which correlates with areas of differential hemodynamic stress. And with that, I would like to thank Dr. and Mrs. Ellen Gogan, the program administrators, my mentor and lab members, and here are my references. Thank you.